In many living organisms, glucose is a significant source of energy that must be used efficiently. The regulation of glucose is important to maintaining overall health. In humans, two main molecules serve this function: insulin and glucagon. Today we'll be focusing on insulin, which is a molecule that is used as a signaling device to allow glucose to enter the cell for storage. Insulin is composed of two polypeptide chains, known as the A chain and the B chain. These chains are linked by two disulfide bonds. Dimers usually form between the C terminals of the B chains due to hydrogen bonding. However, in the presence of zinc ions, these dimers can join to form hexamers. Insulin synthesis occurs in the beta cells of the pancreas in response to high levels of glucose. First, the insulin mRNA is translated as a single-chain precursor called pre-proinsulin. The removal of one peptide during insertion into the endoplasmic reticulum creates proinsulin. Proinsulin is a precursor of insulin that has three domains: a carboxyl terminal A chain, an amino terminal B chain, and the C peptide, which is what holds both chains together. In the endoplasmic reticulum, multiple endopeptidases cause the removal of the C peptide, thus creating a mature form of insulin. Insulin is packaged into secretory granules in the Golgi apparatus. Then, the insulin diffuses into the small blood vessels of the pancreas, where it is ready for use. When blood glucose levels rise, insulin from the pancreas travels down the bloodstream to a cell. Insulin activates the insulin receptor, tyrosine kinase, which is found in the cell's plasma membrane. A phosphorylation cascade is created that transfers the signal down the pathway. One of the last proteins in the signaling pathway moves away from the plasma membrane towards the GLUT4 storage vesicle pool. When the signal from the insulin pathway reaches the GLUT4 storage vesicle pool, the vesicles are released from their resting state and move towards the plasma membrane. The GLUT4 pool will then merge with the plasma membrane, causing GLUT4 proteins to emerge on the membrane itself. This leads to a great increase in the amount of glucose taken in by the target cells. The glucose can then be used for glycolysis, used in the production of triglycerides, or stored as glycogen. Diabetes increases the risk for insulin resistance, which is when the insulin receptor does not react to the binding or presence of insulin outside the cell. There is no phosphorylation cascade, so the GLUT4 proteins do not bind to the plasma membrane. Without these proteins, glucose is not taken up as efficiently by the cells, so it builds up in the blood. The lack of energy in the cells can cause many detrimental side effects, such as increased fatigue, headaches, and excessive hunger.